Welcome to the RSCBelgium section. My name is Tim Reynolds. I'm chair since about January the 17th. Again. Anyway, so welcome to the first talk of the season. And this evening we have a valedictory lecture from our past president, our past chair, Bob, who has just completed is it 6 years as chair? Something like that.
I thought term 6 years, yeah.
It was, yeah. And he'll be talking to us about gaming theory and the 2024 Nobel Prize for Chemistry. I shall introduce Bob, although most of you will know Bob from previous encounters. But Bob Crichton graduated in biochemistry from the University of Glasgow in 63 BSc and 66 World Cup winning year as a PhD. And after carrying out postdoctoral research at Max Planck in Munich, he was appointed as a professor of biochemistry at the Universite Catholique de Louvain in Louvain-le-Neuve in 73, and I think he introduced biochemistry as a mandatory part of the curriculum for all chemists at the university.
A condition of my appointment.
Yep. And his main research interests, of course, are in the inorganic biochemistry of iron and related metals. Bob's achievements in biochemistry of iron proteins have been recognised at international level, and is a worldwide leading figure in this important area of science. He's now Emeritus Professor, and he's been the author or co-author of some 200 scientific articles over, and more than 15 books, I'm thinking now, probably 60s and coming on for 20, including some best scientific sellers, so to speak. From the perspective of the Belgium section of the Royal Society of Chemistry, Bob gave the section's inaugural lecture, I think.
Yes, the first lecture was in a cafe on the Mont des Arts.
Yeah.
On the future of biotechnology or something, I think, protein biotechnology.
Yeah. And he's been obviously a regular member of our committee for most of his existence since the early 90s, and he has had 3 periods as chair of the section. Tonight he's going to talk to us about the background of the 2024 Nobel Prize, and so over to Bob.
Thank you very much, Tim. Right, I shall try, in the interests of my cameraman, not to move around too much, but this is not in my habit when I'm giving a talk. I usually wander around and sort of talk to the audience and try to see if people are following what I'm saying rather than sort of looking at the screen all the time. Anyway, I have to say that this was supposed to be a café chimique, so there won't be any cafe, but there'll be drinks afterwards. The chimie you're going to get now. And it's going to be basically telling you how somebody who was a specialist in making games for computers became one of the pioneers of predicting protein structure, and together with a colleague obtained the half of the Nobel Prize for Chemistry in 2024, together with another colleague from Washington who got the prize for making proteins which he was predicting the structure of and predicting the properties of them. So he was making proteins to do specific functions, starting from just a sequence of amino acids. Anyway, next slide, please. The proteins are, in fact, the machines of the cell. Nothing much in a cell happens without proteins being involved. They're made up of amino acids. There are in general 20 of them, and these form a chain of amino acids joined by what we'll see in a minute are peptide bonds, and they form a 3-dimensional structure. And as we will see, one of the challenges of modern biochemistry is can we predict the 3-dimensional structure of a protein simply from its sequence of amino acids. As I said, they're the structural building blocks of things like muscle, feathers, all sorts of things. They are involved in being antibodies and hormones. They are enzymes which catalyze all of the chemical reactions that go on in a cell. And they are involved in signaling and in communication channels. They're all over the place. Next slide, please. Now, I'm just going to show this slide very briefly because I'll go into detail later on. that we recognize 4 levels of structure in a protein. There's the simple amino acid sequence, which is the primary structure. There's the secondary structure, which is made up of local arrangements of the polypeptide chain to form helices and to form sheets. And we'll talk about those. These exist in the structure and are usually connected to one another by loops, which are not so ordered. Then there's the actual 3-dimensional structure of the protein, which is called the tertiary structure. So we've got primary, secondary, tertiary. And then there are a number of proteins that actually have more than one subunit, and they have what we call a quaternary structure. As you will see, one of the proteins that we're going to look at in detail is haemoglobin, which is a tetramer, and which we'll see has some very important properties which are conferred on it by the fact that it's tetramer. Right, next please. So, a very important experiment was carried out by a good friend of mine at the NIH, Chris Anfinson, back in the 50s and early 60s. He must have done this experiment dozens of times because he wanted to be absolutely sure that there was no other interpretation of it. And his experiment was very simple. He took a small peptide, a small protein enzyme, which was called ribonuclease. Ribonuclease is an enzyme that breaks down, as you'd imagine, nuclease. It breaks down nucleic acids. Ribonuclease, it breaks down ribonucleic acid. Proteases break down proteins. Lipases break down lipids. Glycosidases break down carbohydrates. So it's a nuclease, and it's a single chain connected by 4 disulfide bonds. There is one additional covalent linkage that you find in proteins, where you have 2 residues that have sulfhydryl groups, cysteines. They can join together to form a disulfide bridge, which are shown here just as the connections between those. Right, so what he did was he took his ribonuclease, which is fully active, he put it into what we call a protein denaturant. This is a solvent that unwinds proteins and allows the hydrophobic parts to get comfortable with the urea, which is the solvent. And he added mercaptoethanol, which is a thiol, which could reduce the disulfide bonds to sulfhydryl groups again. And then you get an unfolded, reduced ribonuclease. And this, I do not need to tell you, has no biological activity whatsoever. Next slide, please. What he then did was he took this and he dialyzed it. So he put it in a semi-permeable membrane, let the small molecules dialyze out, leaving the protein behind. Then he oxidized it in the presence of molecular oxygen, and he got back fully active ribonuclease. Now, if you've got 8 thiol groups in a protein, there are 105 different ways that they can combine together. And yet it's only one of them that forms. And so Anfinson came to the very reasonable conclusion that all of the information that a protein needs to form its 3-dimensional structure is contained in the amino acid sequence. So, if we could guess how we go from an amino acid sequence to a 3-dimensional structure, we could save all the hard labor that you will hear a little bit about from the point of view of Max Perutz and John Kendrew, who spent years determining the structure of the first 2 proteins to have their structure determined by X-ray crystallography. That's myoglobin and haemoglobin. We also crystallized some proteins and did some protein structure. Believe me, it's a long and arduous task. And if you could do it by just slotting in a computer program and saying what is the 3-dimensional structure, it would be marvelous. Next slide, please. So here you have a very nice photograph taken in the Cavendish laboratory. Well, it's taken at the MRC in Cambridge, the MRC Molecular Biology Lab. Max Perutz on the left, John Kendrew on the right. Max Perutz with the balsa wood model of 5.5 angstroms resolution of haemoglobin, and Kendrew, as you'll see later, with the stick model. You've got a grid 1 centimetre to an angstrom across the bottom, you've got poles coming up every angstrom apart, and you stick the amino acids onto that on the basis of their coordinates, and you make a three-dimensional structure. He's standing with a model of myoglobin. In fact, Perutz started in 1936. He left Vienna in 1936. His father was a reasonably wealthy industrialist, and he was able to go to the Cavendish and get a job working there with Sir Lawrence Bragg on the structure of haemoglobin. And within a year, the year after he arrived, 37, Dorothy Hodgkin, who's the doyen of crystallography for most people, and J.D. Bernal were able to show that proteins, these big molecules, were able to be crystallized and give beautiful X-ray diffraction patterns, which meant, in principle, that it should be possible to determine the three-dimensional structure of protein from its X-ray diffraction pattern from the crystals. They came back to the Cavendish after the war. Obviously, Perutz got sent to Canada because as an Austrian, he wasn't regarded as a very good security person. In contrast, Kendrew spent his war working in intelligence, first of all in Egypt and then in Asia, and he came back with the rank of flight lieutenant in the RAF. But he joined the Cavendish and started working together with Perutz on the structure of proteins. And at that time, as I say, in 1950 thereabouts, they actually established this unit, which they gave the extraordinary name to, which is a department for research on the molecular structure of biological systems. When they subsequently moved, after seeing a lot of other things taking place, because this institute was the home of a whole lot of people working on the structure of DNA, on muscle contraction, on the structure of viruses, and it was in fact they moved in 1960. In the beginning of the 60s to the laboratory of molecular biology. And when I came here to Belgium in 1973, we were still arguing about whether biochemistry was really the only thing that existed. My head of department in Glasgow, J. Norman Davidson, who was a nucleic acid man, he said, molecular biology, the practice of biochemistry without a license. Well, it wasn't in Cambridge. They called themselves molecular biologists from 1960s. So let's be fair with it. About molecular biology was not suddenly a newbie that came in to get funding, because there's a tendency to find a new name if you're looking for some funds. Always helps. In 1951, Max was giving a very important lecture to an assembly at the then Cavendish laboratory. And he was talking about his structure of haemoglobin. He was very proud of this, and the audience was full of all the major protein chemists in Britain at the time. And when he finished his talk, he was followed on the programme by one of his PhD students, whose name I'm not going to mention at the moment. This PhD student took up and gave a talk which he entitled, "What Mad Pursuit," and proceeded to tell the audience that the structure that Perutz was presenting was complete nonsense, and that there was no way in which X-ray crystallography was ever going to be able to solve the structure of haemoglobin. I'll come back to that later. Actually, Bragg wanted to sack the student in question, but Perutz said, no, let him stay, because he's talking a bit of sense, actually. And it turned out that during his meandering talk about how much rubbish it was and the structure and all the rest of it, he did mention one way in which it might be possible to determine the structure of proteins. My professor of physical chemistry in Glasgow was James Teith Robertson, And Robertson had developed a method for solving the structure of small organic molecules that were a bit tricky. If they were small enough, only a couple of dozen atoms, you could guess the phases, and you could do a Fourier transform and get the diffraction data. But with a couple of thousand atoms in a protein, there's no way you could do that. And what Monteith Robertson had shown was that if you took a heavy metal, now I can assure you in the 1950s and 60s, heavy metal wasn't yet invented as a rock group. You might have heard of them in that context. But heavy metals are things like mercury. And what Robertson showed was that if you incorporated a heavy metal atom like mercury into the small molecule without changing its structure, then you could take an X-ray diffraction pattern and you would see the very intense electron density of the mercury atom, and that would enable you to determine the phases. Because the problem about X-rays, bloody nuisance, they don't go in straight lines, they're in waves. And if you don't know which bit of the wave you're in, you can't synchronize your data. So you need to know that. And that was the so-called phase problem. And the method of isomorphous replacement, where you replace, you put a heavy metal into the molecule, and then you see it without changing the structure when you do your collection with and without the metal, and then you solve the phases. So the other thing which was quite unexpected was a colleague, Austin Briggs at Harvard, actually discovered that you could introduce mercury into sickle cell anemia, haemoglobin, without changing its properties, which told us that you could introduce mercury into hemoglobin with isomorphous replacement. So next slide, please. Now I have to be careful with this slide. This slide comes in two variants. There's this one, and then there's the second version, which I'll tell you when to move to. So don't be surprised if it looks like it's the same slide coming up twice. I'm not going potty. There's a reason. Perutz managed to crystallize hemoglobin with some mercury salts in it, and he was able to collect data. And when he went down to the dark room, the dark room's always on the ground floor in the basement, he had all the way down to the third floor. He'd gone to he developed the pictures of the X-ray diffraction photographs, and he could see that in the one with the mercury there were intense spots that weren't there in the other one. So he rushed up the stairs to Bragg's office and said, "You must come down to the dark room. We've got it." And when Bragg saw it, he said, "Yes, indeed, this is going to enable us to solve the phases." And in his great enthusiasm, Perutz suggested that he might be able to do it in a year. It took him another 6, but never mind. That's the way it is in X-ray crystallography. In the meantime, Kendrew decided to do the crystal structure of myoglobin. Now, myoglobin is a smaller protein than hemoglobin, and it's found in muscle, where it stores oxygen, whereas hemoglobin transports oxygen, picking it up in the lungs and delivering it in the tissues. Myoglobin stores oxygen in the muscle, which can then be released to hemoglobin when it's required. And he tried all sorts of species, because that's the other problem. I didn't tell you this. You didn't buy your proteins off the shelf from Sigma Aldrich or from Boehringer. You went down to the abattoir and you got the appropriate material that you needed to purify it from. We used to send people regularly to Charleroi on a Thursday, because that was when they slaughtered the horses, and we could have the horse spleen to make our ferritin for free. We usually, to be kind to them, we buy a few horse livers, but so you had to start with it. And he tried with all this stuff, and he eventually discovered that he could get superb crystals of myoglobin using sperm whale. And it just turned out that in Cambridge there was a low temperature research station, and they had tons of the bloody stuff sitting around in their cold room. So he just would walk down the road to the low temperature station, pick up his bucket of ice full of muscle from the sperm whale, and purify the stuff. and he was able to incorporate, again, mercury into it and to solve the phase. And as he describes in a wonderful article he wrote for the Scientific American, he said, there are moments in history when you come over the horizon like the captain of a ship and you see ahead of you something which nobody has ever seen before. That was our joy on a Sunday morning in Cambridge when we saw for the first time the three-dimensional structure of a protein molecule. It was at low resolution, but it was still a structure. And he went on to get the structure at a resolution of 2 angstroms and eventually 1.4 angstroms, so atomic resolution. In the meantime, Kendrew and Perutz published a paper showing that they had actually... I must come back to the point I'm going to make in a minute, yes. Just want to make sure that... Right, yes. Kendrew and Perutz published a paper where they compared myoglobin and haemoglobin from a number of vertebrates, because one of the advantages of evolution, and I'll come back to the importance of this later in the talk, is that if bits of an amino acid, if amino acids are interacting with one another in the three-dimensional structure, they will have to be conserved in evolution. So if one of them changes, its partner will have to change in the same way so that they can still interact. That is an important thing. So they were comparing, and they found that comparing haemoglobin and myoglobin for a series of vertebrates, only 9 out of more than 1 to 40 sites are occupied by the same amino acid residue in all the globin chains. They did have many things in common, and the thing that was in common, we'll come back to that later, is that they have the same characteristic three-dimensional folding of the polypeptide chain, which we call the globin fold, but I'll come back to that. So, Kendrew and Perutz were jointly awarded the Nobel Prize for Chemistry in 1962, and when they went there, next slide please, this is the same slide, but with the photograph of the gentleman who had interrupted him in 1951 and told him he was talking rubbish. This is Francis Crick, who by coincidence got the Nobel Prize for Physiology or Medicine in 1962, together with 2 other scientists who we will have heard of, Watson and Crick, and of course Wilkins, who had actually not given the X-ray structure of his student Rosalind Franklin to Crick and Watson. He'd shown it to Perutz when Perutz was on a visit to the MRC unit at King's College that they were in, that Wilkins was in, and Franklin was still alive at that time. You know the classic story, Nobel Prizes cannot be awarded posthumously, so Rosalind Franklin didn't get the Nobel Prize for her pioneering work on the X-ray structure of DNA. It went to Wilkins because he was her boss and he had inherited her program when she died. But anyway, it was Kendrew who had been the student who'd been rude enough to tell Perutz. So you can see how clever a boy Francis Crick was. Already as a PhD student, he was able to see the problem that lay ahead of Perutz in thinking that he could just take his lovely X-ray photographs of haemoglobin and solve them. So let's go on. By the way, for those of you who are interested, I have put at the bottom of the slides references if you want to go and look up the information in the published papers. That's an X-ray photograph of a myoglobin crystal, and this is a structure of the 5 angstrom resolution structure, the low angstrom structure. We'll analyse it in detail in a minute. And this is Kendrew in the process of making this map. Well, we made a model of lysozyme in the lobby of the university. I can remember in Glasgow where before I left, lysozyme was one of the first enzymes to be crystallised, and you had to make this ridiculous thing. The workshop would make a big grid, and then poles coming up, and then you would take the coordinates and laboriously plot them out and fix them onto the structure, and you would then build this ridiculous looking thing here. And of course there is one thing that characterises, this is the ribbon diagram, which is the sausage diagram, This is the ribbon diagram where you just show the helices. And you will see immediately that this protein is entirely alpha helical. It's got nothing but helices. And there's an extraordinary graphic artist called Irving Geis, who for many years used to illustrate articles in the Scientific American and in textbooks. And this is his drawing to scale of the three-dimensional structure of myoglobin in its atomic detail. And what you can see here is something which is very important. If you were looking for similarities between two proteins, you'll see them a damn sight easier in that diagram or that one than you will in this, because here you will get lost in the detail. But if you just have a 5 angstrom structure, the backbone stack stands out and you can see how similar they are. So next slide, please. This shows you hemoglobin. And here's the hemoglobin tetramer. And in fact, the hemoglobin tetramer binds oxygen. And when it binds oxygen, there's a mechanism which moves this helix here, the F helix. The blue is the deoxyhemoglobin, which we call the tense state, and R is the relaxed state, which is the oxygenated form. And the fact that this transition occurs in a tetramer means that whereas the first oxygen has great difficulty getting onto the protein, the next 3 get on more easily, and the 4th oxygen on binds with a binding affinity 100 times more than the first one. That's what we call cooperativity. And it's only possible because you've got a tetrameric structure. It doesn't work with a monomer. And you can see here the oxygen binding curve of myoglobin is just a straightforward hyperbole, whereas the binding curve of hemoglobin is a sigmoid. Next slide, please. So this is the globin fold, and I want just to take you through it. It's very simple. You start off with the amino terminus. You've got the A helix. You come down the B helix. You then go through this CD region. Then you've got the next helices, and it all ends up at the C terminus down there at the end of the G helix. So 8 helices, 6 of them stacked 3 by 3, with a little floppy bit out here. Other than that, it's just helices. But what's important is if you take heme and you put it in the presence of oxygen, it will just oxidize the iron 2 to iron 3. That doesn't happen in myoglobin. It doesn't happen in hemoglobin. It doesn't happen in any of the thousands of globins that have this heme oxygen binding site. We don't still understand completely why, but in this configuration, with a histidine linked to one side and a distal histidine which acts to help bring the oxygen here, this oxygen is protected from being transformed, and the iron remains ferrous, so you can get oxygen binding without changing the oxidation state of the iron. And that's a trick which, once you've found it, you better be sure that nature is going to keep it, which is why we find the globin fold in not just in mammals, we find it in plants, we find it in invertebrae. When I was working in Munich, we used to purify Chironomus tumi hemoglobin from insect larvae. Chironomus is a fly that you find in the lakes in Sweden. And you can buy the larvae as a bait for fishermen. Yeah. It comes as a mucky red paste, and if you extract it, you get almost a pure solution of hemoglobin, and there are 12 different hemoglobins in there. What we did during my year in Munich was to separate all 12 hemoglobins, and we published a paper, Uber Chironomus hemoglobin tumi. And then when I left, some clever clogs called Boozer came in behind me. The name tells you it all. and he published a paper with Braunitzer in which he said, eine verbesserte Trennung von haemoglobin. This was supposed to be a better purification. It was a bloody useless preparation. But we got all 12 separated. And one of them crystallized, and our neighbour across the courtyard in the Max Planck Institute for Eiweiß- und Lederforschung, protein and leather forschung research, Robert Huber, determined the structure of chironomous chemoerythrin and showed that it had the globin fold. So it was another hemoglobin, and that established this whole idea that the globins were a family that you find all over the place. Next slide. Now, I said the primary structure is the sequence of amino acids. This is the sequence of insulin. It's made up of two chains, an A and a B chain. The A and B chains are connected by inter-chain disulfides. There's an intra-disulfide bond in the A chain. The secondary structure, I should have, just while I'm passing by, it turns out this is Pauling. Pauling was able to determine the amino acid, determine the X-ray crystallography, a whole lot of small peptides and amino acids with Corey, and he showed that all of the 6 atoms of the peptide bond, forming the peptide group here, there's a peptide bond here between oxygen and carbon and nitrogen. All of the amino acids, all six residues are in the same plane, which means that if they're all planar, if you know the angles here and there, you can tell where the chain is going to go in 3 dimensions. And this, here's I quote, I have to put glasses on for this, excuse me. This is Linus Pauling. He was in April 48, he was a visiting professor in Oxford, and he caught a cold and it was damp outside. And he got fed up reading detective novels. So as he said, I took a sheet of paper and sketched the atoms with the bonds between them, and then folded the paper to bind one bond at right angles, what I thought would be relative to the other, and kept doing this, making a helix until I could form hydrogen bonds between the turn of the helix and the next turn, and it only took a few hours of doing that before he discovered the alpha helix. The alpha helix disappointed the purists, thank goodness, because it doesn't have an integral number of residues per turn of the helix. It has 3.6 residues per turn of the helix, but it forms a very beautiful linear hydrogen bond between the CO of one bit and the NH further up. And you can see here the way this works out. If it's between the first oxygen and the second nitrogen, it's a 2-2 ribbon. For the third, it's a 310 alpha and pi. And the 310 helix you find often at the end of an alpha helix, because the 310 helix allows the chain to turn through 180 degrees. So it means you can go off in a different direction. But he discovered this long before we had this X-ray structure of any protein, just on the basis of looking at dipeptides by crystallography. The other one that you find inclusive the helix is the alpha helix is by far the most common. But the other one you find are the beta-pleated sheets, which can be local, anti-parallel. So if you've got a chain that comes along and then you've got a bit that just turns around, then they can form 2 anti-parallel pleated sheets. But bits distant away in the whole amino acid sequence can form parallel beta sheets, which are a little bit different in shape, not as nice hydrogen bonds as in the anti-parallel. But these structures are well known in proteins. Next, please. This just tells you a little bit that I want to remind you important about the tertiary structure. In the tertiary structure, you have several kinds of interactions. Polar amino acids that are slightly polar or charged, will tend to form either ionic bonds or hydrogen bonds between the polar groups. On the other hand, hydrophobic amino acids, which are very sticky carbon-rich chains, will interact with one another and exclude water to make themselves feel comfortable. Because remember, the only reason that we are living on this planet is because we are water. So we live in a watery environment. We didn't. If ice didn't freeze the way it does, there would be no life in our lakes. You might not have noticed, but ice doesn't freeze from the bottom up, it freezes from the top down. And it's lighter than water, so it floats. Okay, these are just two other structures, of three-dimensional structures of proteins. This one is triose phosphate isomerase, which is an enzyme in intermediate metabolism, and it's made up of a mixture of beta-alpha beta structures. Whereas this one, the superoxide dismutase with its copper and zinc, is in fact pure beta sheet. It's just one series of beta sheets after another. Right, next slide, please. Now, way back in the 1960s, when haemoglobin and myoglobin were having their structure determined, the number of protein structures available was very, very small. So making any kind of comparison with, starting to make predictions when you've got hardly any structures is not very easy. This is the protein data bank set up around this period, and this is the number of experimentally determined protein structures deposited in the protein data bank, and you can see that it's increased enormously. So we now have a much bigger database, and all of these proteins will have all of their atomic coordinates deposited in the database. So if you want to use them for any purpose, you can get access to them. But this just shows you how very little was known early on. Next, please. So, as I said, we can see a few obvious things that come out of these early structures. There's the secondary structure motif, which is quite easy to identify, and in fact, Chu and Fassman, using a data set of 15 proteins, which the structure was known, worked out which amino acids were most likely to be found in helical structures or in beta sheet structures. So you could then start making predictions on what would be the likely secondary structure of the polypeptide chain. That, of course, ignores a very important point, because the tertiary structure is not just going to be held together by the alpha helix and the beta sheet. It's going to be held together by the interactions between amino acids in those structures and between those structures. And that is not so easy to get at. And you'll see that that's where a bit of sleight of hand comes in, shall we say. Next slide, please. The other things that became very clear is what we call the hydrophobic effect. I said to you, we live in a watery solvent, and water is polar. The oxygen in water wants to make hydrogen bonds with the protons and other water molecules. And so amino acids like valine and leucine, which have methyl groups at the ends, long methyl chains, they don't like being in a watery environment. So what tends to happen is that all the hydrophobic amino acids collect on the inside of the protein, and all of the polar residues collect on the outside. But when you get polar residues on the inside, they will form interactions, either ionic bonds or hydrogen bonds, with other polar residues. That's important because, as we'll see later, one of the things that came out when you start trying for ways to get computers to look for data is to say, if you've got amino acids that are close to one another in the three-dimensional structure, and you look at a whole series of organisms, the same protein in all these, if these are essential for the structure, when one of them gets changed to from a polar to a hydrophobic, its partner will need to go hydrophobic as well. And if it does, then it's kind of confirming that these parts of the structure must be close together. This is just one of the little lead-ups that's coming later. Okay, now we have a huge number of DNA sequences as well. I mean, since I used to determine the structure of proteins, which took forever, you now simply take the cDNA, you sequence the DNA, and you can write the amino acid sequence of the corresponding protein. Toffee. In fact, we know about 3 billion structures, DNA sequences. And the number of protein sequences that have been so far identified is over 200 million. But in fact, up until quite recently, the protein data bank contains only a total of about 200,000 proteins for which the X-ray structure of the NMR structure has been determined. Next, please. Now we come to the first of our Nobel laureates. Very strange. I was having lunch with with Bobby's family on Sunday, and I was speaking to her son-in-law, and he said, "Demis, yes, of course. He's a North Londoner." And he started off. It's quite interesting. His parents were Greek and Singapore. Born in North London. He was playing chess at 4. He was a grandmaster at 13. He went to Barnet, Queen Elizabeth School, Barnet, for a couple of years, and then his parents took him out and home tutored him. And at the age of 16, they sent him to Christ Church College in Finchley, where he did his A-levels at the age of 16, and was accepted immediately for Cambridge to do a degree there. But Cambridge suggested to him that he was a bit young to come to Cambridge. So he spent his year working for a colleague who had a computer games company, and he actually devised, he won the job with Bullfrog Productions by entering an Amigo Power win a job at Bullfrog competition. At 17, he co-designed and lead programmed the game Theme Park, and he earned enough money from his gap year to be able to pay his way through university. That's not bad. When your kid wants to say, I fancy a gap year, tell him, how about you earn enough money during your gap year to pay for your education? This would be good. He then started a separate outfit developing games, and then he sold it off and started studying neuroscience, initially at University College London, and then in the States, and he finally got his PhD from UCL in 2009 with a thesis that was entitled, if I read it correctly, Neural Processes Underlining Episodic Memory. And he published a series of very interesting papers in journals like PNAS, Science, Nature. Yeah. Next slide, please. He used what he'd learned about brain to develop better neural networks for AI, and in 2010 he funded the company DeepMind in London, initially making board games and using them as a proving ground for AI and developing AI models for popular board games. The mission of AI, as is the mission now of the Chinese company that have just knocked the bottom out of ChatGPT, is to develop artificial general intelligence, which is a computer that will be able to think, to solve any problem you give it as well, if not better, than a human. That's the avowed objective, or that was the avowed objective of DeepMind. And although the company was doing quite well, he sold it to Google in 2004 for a modest £400 million, and kept DeepMind for himself, leaving the health part of it to be incorporated into Google Health. He then developed these programs which allowed him to solve board games, two-person board games like Go, like chess, And he eventually was able to develop programs that were good enough to beat champion Go players. And in fact, in the match against the one poor South Korean chap, he actually won 4 games out of 5. The computer did. And in one move of the game, it actually used a move that had never been seen in people playing Go, which suggests that if you've got a huge amount of information available in the computer, it can think beyond what a human brain would be able to do, which is an interesting hypothesis. However, for Demis, the goal was not to develop better AI, but was to develop better AI models. And using his knowledge of neuroscience, he started to do this and decided to enter a competition to predict protein structure. Next slide, please. That's in 1994. The CASP experiments, critical assessment of protein structure prediction, were created by John Moult and Fidelis. And these annual challenges, they would send out a series of amino acid sequences of proteins that they knew were going to be published as structures. And before the structures were published, the predictors had to send in their predicted structure. Mm-hmm. And then they were compared. And in fact, in the early programs, they were able to get to about 40%, never anything better than 40%, which is not very much use. So nothing much was happening until we get to the CASP competition, CASP 13 in 2018. We can have the next slide, please. This just reminds us that in 2018, DeepMind decided to start devising a computer program that would try to solve the problem of predicting three-dimensional structures of proteins. And this program was called AlphaFold. We now call it AlphaFold 1. And initially, it was done by using genetic information, by isolation, by analysing covariation between structures that were homologous, usually from different sources. So you could see which amino acids were likely to be in contact with one another. And as I said, you found then the prediction would be that if you could then make a distance map between the residues and start going on from there to try and get a structure. The algorithms and things that they used were basically training a neural network to make accurate predictions of distance between pairs of residues, which can convey more information about the structure than simple contact predictions. Using this information, they were able to calculate structures, and the program eventually achieved quite high accuracy in 2018. Next slide, please. This shows you where CASP was at until 2016. 2016 they got to 40%, and then suddenly in 2018 it jumped up to nearly 60%. Now this is getting very encouraging, and so that was the project, the projection, the progress of the thing, and this was their performance relative to the other 15 top entries in CASP 14, which we'll come back to in a minute. But I'm not going to go on with that. Let's go to the next slide, because we're going to meet the next persona. Next, please. The next dramatis personae is John Jumper. Jumper was going to do. Sorry, we've got one. I think. Hang on. Well, yes, okay. They, as I said, they got to 60%, but by that time they were not able to get AlphaFold to do any better than that. And that was the point at which Jumper joined the team. Now, he started in university to study physics and mathematics. And while he was at university in 2008, he was in the unusual position that he started getting interested with a company who was using computers to simulate proteins and their dynamics, and so he decided to do an MPhil in physics at Cambridge, and then went on to do a doctorate at the University of Chicago. And in 2017 he heard rumours that DeepMind were starting getting interested in protein prediction. So he applied for a job and was hired, and the rest, as they say, is history. Because, as we'll see in the next slide, this was the real breakthrough. This was AlphaFold 2. And in AlphaFold 2, I'm not going to go into this in great detail because A, I am not a computer scientist, and B, the language is quite complicated and I'm not sure that I understand it. But anyway, what they basically tried to do was to take into account interactions that were taking place between amino acid residues that were close in the tertiary structure, but not necessarily obviously close. And the way they did that was to take a whole family of animals, other species, that had homologous proteins, and to compare them, and then to see which residues were conserved all the way through, or which, if they changed, resulted in a change in what would be their corresponding partner. And this information was built in. It was all very complicated, and I'm not going to go into the detail because it's there for you to read if you want to. But let's move to the next slide, which is just the... So the main modules of AlphaFold 2 were what was called the evoformer and the structure module. Now I'm going to actually go on 2 slides and then come back if I may. So I'm going to go to the next slide, please, and show you the 4 steps of how AlphaFold works. In the first step, you take the amino acid sequence of the unknown structure, you introduce it into the AlphaFold program, and you search the databases for similar amino acid sequences and structures. The sequence analysis, then, the model aligns all the possible sequences from the different species and investigates which parts have been preserved during evolution. In the next step, it explores which amino acids could interact with each other in the 3-dimensional structure. Interacting amino acids evolve, as I said, if one is charged, and the other will have to have the opposite charge so that they're attracted to one another. If one is replaced by a water-repellent hydrophobic residue, the partner will have to be replaced by a phobic hydrophobic as well. Next slide, please. This then shows you the artificial intelligence analysis. Using an iterative process, AlphaFold refines the sequence analysis and distance maps, uses neural networks called transformers. The great thing about a transformer, apparently, is that it indicates to you what are the important elements to focus on. Data about other protein structures, if you found those in step 1, are also utilised. And then finally, AlphaFold puts forward a puzzle of all the amino acids and tests pathways to produce a hypothetical protein structure. It reruns this through step 2. After 3 cycles, it arrives at a particular structure down here at the bottom. And then an artificial intelligence model will calculate the probability that different parts of the structure correspond to reality. Then you have your so we go back up. That just shows you the 2 slides. Yeah, this one next down. That's it. The one that's just after that, please.
Oh, after.
Yeah, that's it. Yeah. Oh no, after we come going back. That's it. Yeah, that the one that was before the yeah, that's it. Yeah,
This one.
that one. So this just shows the 2 modules. The input data with the sequence structure database serve as the input to the evoformer, and the structured module then produces its output, a 3D model of the protein structure. And it takes each peptide bond as a triangle, and it floats these triangles around until it gets them to fit together. And then you come up with a sequence with the amino acid side chains in their appropriate positions. We can go on now, two slides further. That's it. Now we come to the last of the 3 recipients who got 50% of the prize, because David Baker also started looking at computational ways of getting to the structure of a protein, and then gave it up and started making synthetic proteins that would do something that wasn't usually being done. He was going to do philosophy and social science, but by good fortune he landed on a copy of the classic textbook Molecular Biology of the Cell. I think most of us, I can see at least one person in the audience who certainly has a copy of that on his shelf, and I do, in my bookcase in Louvain, in Wavre. And this made him to realise that biology was an interesting subject. In fact, it's very interesting. In the 1950s, a lot of physicists and chemists were getting interested in biology because they felt that there was nothing new to find in chemistry or physics. So if there were any new ideas or principles to be found, they were likely to be found in biology. So you find a lot of physicists and chemists moving towards biology as a means of hoping that they would find something new. Anyway, he joined CASP in 1998 using a program that he developed that he called Rosetta. But he eventually gave up doing this and decided it would be more interesting to use his prediction program to make proteins that would have a specific structure. And the first of these he made was top 7, which is on the next slide. I'm going to go through these very quickly. Yep, please. So this is top 7. This is the first protein that was entirely different. It was made just by predicting a sequence, and that's the structure they predicted it would have. Beta sheets with 2 helices. He then cloned the cDNA corresponding to this protein into a bacterial vector, expressed it, isolated it, crystallized it, and showed that the X-ray structure corresponded to the 3-dimensional structure that was predicted, which is pretty impressive. So that's what you see in this slide. Now I'm just going to end the talk with a few simple little illustrations of what he's been doing since then. And one of these you might find a little bit interesting in view of what's going on. Next slide, please. Thanks very much. One of these you might find quite interesting with what's going on at the Mexican and Canadian border at the moment, because he designed in 2017 a protein that binds the opioid called fentanyl. The fentanyl recently in the news? Yes, it's what seems to be flooding across the Mexican border. And he developed a protein that would bind it and which could be used to detect fentanyl in the environment. Quite useful. He also designed proteins that would function as molecular rotors, nanoparticles that could initiate that express influence visors on the surface, so they could be used as a vaccine. Shape proteins that can change their shape by external influences to make tiny sensors. Next slide, please. And a few others. An enzyme that can decompose plastic. It's quite interesting. A bacterial enzyme that causes antibiotic resistance. And a molecular structure of in the human body, which is forms a pore through the membranes surrounding the cell nucleus. Last slide, please. The conclusions are that we've got to a stage where both the structural design and the prediction problems are largely solved. These have enormous implications. The AlphaFold team made large databases, created large databases of predicted protein structures, first for the human proteome, and then for the majority of sequences, the 200 billion that are available on the UniProt source database. This means that almost overnight we got access to orders of magnitude more structural information. Likewise, the protein design field has reached a stage where some of the most exciting projects are to make things like protein-based medicines and vaccines and applications in synthetic biology. So this has large consequences, and I think will continue to have large consequences. I'm going to leave you with a slide which just illustrates that even the Nobel Committee have a sense of humour. I hope you enjoyed the talk, and I look forward to answering your questions. Last slide. Here you are.
Thank you very much, Bob. I'm sure there's a few questions. Does anyone want to kick off? question? Overwhelmed.
We are overwhelmed.
That's all the science of Brazil.
On the computational aspect, it's on the haemoglobin.
Yeah.
What happens with carbon monoxide in haemoglobin?
Carbon monoxide helps to bind to the iron atom of hemoglobin, and it binds in a straight end-on position, which has huge affinity. So it's a very
never be broken down. Is that correct?
Very difficult. Yeah.
That's why you die.
That's why you die.
Yeah.
You simply get rid of your oxygen carrying capacity.
Right.
Very simple. Yeah. It's,
But where do you think this is going to take us?
well,
Because,
I would be interested to know what my dear colleague from Liège, Joseph Marshall, thinks, but I'm not sure that we've solved the structure protein totally. I still think we used to go in the early days in the 60s to protein structure and function meetings, and we were learning more about the structures, and we knew their functions, but what makes a structure give you a function? It's okay, it's easy, it's relatively easy for enzymes, but for proteins that do non-enzymic functions, it's sometimes quite difficult to define how it does it, and I'm not quite sure that we're there yet. Baker is making a lot of progress, but he's making a lot of mistakes, and I don't think we see the mistakes being published. I think what we see are the success stories, which gives the gloss on the picture that everything in the garden is rosy, and I'm not entirely sure that we've solved the problem of why does a particular structure have a particular function, which is really, I think, still quite an open question. Yeah.
Hi. I have a question about solving the phase problem with mercury.
Yes.
Why particularly was mercury selected? Was it because it's a liquid?
Because we have a lot of mercury compounds which can readily bind to thiol groups or to other amino acid residues in proteins. And in fact, in the case of mercury, just happens to be one that was suggested by Robertson, and for some reason people have gone on using it. Quite a lot of people use mercury because of heavy metal. But you can use platinum, you can use other things. Yeah. It's just that, I mean, in the case of myoglobin, Kendrew could never get it to bind covalently to the protein. He had to just diffuse it in and hope that it stuck somewhere and that he would see it in the X-ray photographs. So it was just a, I don't think you could use any other heavy metal, but maybe mercury is easy to get hold of, and mercury salts are quite readily available.